Okay, so welcome everybody to today's seminar. We have here today Juan Pascual and Pedro Costales. They are two PhD students that are going to present their PhD plans. First is Juan is going to start. He is working here at EBD. He has started the PhD last year, so he has 1 year of PhD. He is supervised by Tim Cerdá from the EBD and Francisco Azcárate from the Universidad Autónoma de Madrid. He did the biology degree in Madrid also. After that, he did the master at the University of Pablo de Olavide, and he has been working around at the EBD for several years before starting the PhD. And he is going to talk about how the fire impact ants in Mediterranean ecosystems. So, welcome, Juan. The floor is yours.
Okay. Hello, thank you everybody for coming. My name is Juan Pascual, and for the next 20 minutes I'm going to be presenting my thesis plan called Fire Effects on Mediterranean Communities from an Ecosystem Perspective, which is supervised by Tim Cerdá from here, Biological Station of Doñana, and Francisco Martín Azcárate from Universidad Autónoma de Madrid. I'm going to skip my introduction because it's has summarized it very well, and I'm gonna introduce my research topic. As you may know, here in Spain, we have had several incidents concerning fires, like these ones that I'm showing to you. The first one is in Tenerife in 2023, and these two are from Andalusia and last summer. If we go to the record, we can see that even the years where there are less incidents of fires, there are still more than 6,000 fires every year. Here we have a graph where we can see the affected area by wildfires, and we see that it seems to follow a cycling trend, but what I would like to highlight here is that there are more than 100,000 hectares burned on average per year. What all this together indicate us is that fires are a high frequent and very relevant disturbance in our ecosystems that we must pay attention to. Which are the effects of fire on the ecosystems? First, they promote habitat heterogeneity by opening the habitat. They also change the resource availability, so affecting the nutrient cycling. And also they have a very strong impact on the species composition because they kill a lot of individuals, which allows the regeneration of the old species and the entrance of the new ones. We also must take into consideration that fires have been acting on Earth for billions of years. And then we must understand them as an ecological and evolutionary driver that has been shaping the animal and plant communities and the structure and diversity of ecosystems. In the Mediterranean ecosystems, species have evolved fire-adaptive traits to allow them to persist and thrive in these fire-prone landscapes. Here I bring two examples. The first one is the Pinus pinaster tree, which has cones that need to be heated by the high fire temperatures in order to open and release their seeds. And the other example is the Quercus cruciferous shrub, which has a very strong root system that allows it to resprout after the fire, even when the aerial part of the plant has been burned. As we have explained, these traits give the plant community high resilience to fire. We talk about ecosystem resilience when the ecosystem and its communities have the ability to recover and regenerate after disturbance without losing their functions and services. This has been very well reported in for plant communities, but little is known about how other organisms are doing with fire, like ants. Ants are a very abundant, diverse and conspicuous group in our ecosystems, and they help provide essential services for the ecosystem, like seed dispersal, organic matter decomposition, nutrient cycling, soil movement. They also regulate the invertebrate communities, and they pollinate. Then, understanding how the ant community responds to fire will allow us to have a better understanding about how the whole ecosystem can behave after the fire. And this is the context of my thesis, which has as a main objective to assess the fire effects on the taxonomic and functional diversity of ant communities and their ecological functions. I have structured my thesis in 4 chapters. Each chapter has different specific objectives, and I will presenting them to you during this talk. Let's go with the first one, which is called Impact of Fire Disturbance on Ant Functional Diversity and Ecological Functions, a Global Meta-Analysis. Although the impact of fire on ant abundance and diversity has been very well reported, very little is known about how fire impacts other ecological roles of ants. My specific objective in this chapter is to know and determine the total effect of fire on the ecosystem services provided by ants. And to do so, I plan a meta-analysis. I have followed the Prisma Ecoevo approach. First, I perform a systematic search in these databases, and I have used this query string that includes the keywords fire, wildfire, burn, and ant, and excluded the terms fire ant, solenopsis, and algorithm. Here there is a flow diagram of the work that I have done so far. The bibliographic search, give us back 1,350 studies, and after applying my inclusion criteria on the title and abstract and also to the full text, I have finally selected 203 studies. Then with these studies, I'm extracting their effect sizes to assess how the impact of fires on the ant diversity, abundance, seed dispersal, functional diversity, and dominance hierarchy. When we talk about dominance hierarchy, we refer to the way the community is organized based on the behavioral dominance and the interspecific competitions. To do this, we are giving a dominance category for each of the species reported by the papers. A species can be dominant or subordinate, and this is made in order to assess how fires affect the subordinate species, this type of species. Also, we would like to assess and evaluate if there are an effect of other factors on the total effect of fires on our response variables. This is the end of my first chapter. The second one is called Responses of Ant Communities to Prescribed Burns in Mediterranean Shrublands, Temporal Changes in Taxonomic and Functional Diversity. This chapter is based on the continuation of a previous project called Thilifo, where my group has been working since 2019. This project conducts prescribed fires in the biological reserve of Doñana. So my main objective is to assess the impact of prescribed fires on the taxonomic and functional diversity of the ant community, and also on the composition. My third objective would be to determine if there is a variation within a year, a phenological variation, and also across years. My first hypothesis is that the differences will be more pronounced immediately after fire, and we expect to have a higher and taxonomic diversity and functional diversity. Then, as the vegetation cover regenerates and the habitat characteristics became more similar to the pre-disturbance stages, we expect to have a decline in the diversity until it returned to the pre-disturbance levels. Our area is located inside the reserve, the biological reserve of Doñana, in the area of Monte Blanco. We have selected 18 plots for our controls, and 14 are selected for prescribed burns. We have already burned 11 of them. We plan to burn 3 more next year. These different burning times allow us to create a gradient of post-fire recovery stages. To sample the ant community, we use pitfall trams. We have installed 2 transects of 15 meters in each plot. We install 10 pitfall per transect, and the pitfall are open for 24 hours. We have conducted 2 different periods of sampling. The first one was done by Vidal Cordero, the previous predoctoral student, in 2021. It was done every month during a year, and it will be used to assess the phenological variation of the ant community. And we have done a second period of sampling during last spring that it will be used for assessing the temporal variation. With the community and the species, we are going to calculate these taxonomic indices. And regarding the functional traits, we are giving to each species 13 functional traits related with the ant fitness to calculate the functional diversity and the community with mean. We are also measuring the NDVI index. To assess and determine the impact of fire on the taxonomic and functional diversity, we plan to use a general linear mixed model, and to assess the impact of the composition, we are planning to use a Permanova analysis. Here I'm presenting to you some our principal results. We see that regarding the number of species, there are more species on burned areas compared to the controlled ones, especially during the summer months. And we see that the ant abundance follows a similar pattern. Regarding the temporal variation, we see that the taxonomic diversity is higher in the first year, but then it declines in the second year, and it became more similar to the pre-disturbance stages. A similar trend it's also seen for the taxonomic richness. Regarding the functional diversity, however, we see that there are no changes between the control and the burn areas. All this together can indicate us that prescribed fires can enhance the ant community diversity while ensuring the ecosystem stability, as the functional diversity doesn't present changes after the fire. This is the end of my second chapter. My third chapter is titled Assessing the Influence of Location on Ant Community Responses to Fire, Identifying the Winners. A little bit of introduction. We need to know that dominance hierarchy is a major factor shaping the ant communities, dominant species can limit the distribution and the abundance of the subordinate ones, but this relation of dominance depends on the period of the day and the temperature. Then the dominance hierarchy is dynamic and context dependent. My first objective in this chapter is to assess whether the fire changes this dominance hierarchy. My second objective will be to assess the fire impact on the trophic interactions of ants. And the third one is to know whether there is an impact of the geographic location of fire in these variations. My first hypothesis is that subordinate species will get favor after fire because they are more thermophilic and better adapted to open habitats than the dominant species. And then we predict them to be able to win more competition interactions after the fire. My second hypothesis is that fire will change the trophic interactions of ants, selecting some of them. And my third objective will be that ant community will experiment an homogenization at regional scale, because separated communities will become more similar to each other. To do this, we have selected 4 different wildfires, 2 in Andalusia and 2 in the central systems. There were all Pinus pinaster forests. And what I'm doing on the field is what is called cafeteria experiments. We present a group of baits to the ant community with different resources, tuna, seeds, salt, water, sugar. Here I would like to note that the mealworm is dead, so we can assess the scavenging behavior of ants, and the cricket is alive, so we can assess the predation of ants. We have designed this to install the bait groups at different periods of time, in the morning, midday, and the afternoon, and we do it 3 times for each period in our selected plots that are 3 for each wildfire. Then, during the experiment, we record the identity of the species that came to the bait, also the number of individuals, the soil temperature, and whether there is an interaction or not. Regarding the interaction, we record if it is a competition when 2 different species fight for the bait, if there is a coexistence, whether both species recognize each other but they decided not to fight but share the bait, or whether there are no interaction between the species. And then we plan to do a co-occurrence analysis to analyze our data. This is the end of my 3rd chapter, and the 4th chapter is called The Role of Ants in Post-Fire Trophic Interactions and Vegetation Regeneration. My first objective here is to know whether the fire impacts seed dispersal rate by ants. Our second objective is to know if the identity of the seed disperser ants changes after fire. And the third one is to evaluate if the preference of the ant species changes after fire. This is our experiment that we have designed. Here I would like to note that this experiment is still being designed. So some things can vary in the future, so we can match our objectives. But we have planned for now is that we have selected 2 different shrub species one with elaiosomes, which is Cytisus scoparius, and the other one without elaiosomes, which is Lavandula pedunculata. We are gonna install baits with seeds from the two different species to the ants and observe which species is taking the seeds. So the identification will be in situ, and I have planned this number of replicates in order to have enough data. But as I said, we are still designing this experiment. Before finishing, I would like to highlight the importance of our results. In this thesis, we are following an ecosystemic approach. So by assessing the ecosystem services provided by ants and the impact of fire on them, we will have a better understanding about how the whole ecosystem can behave after fire. And we are doing that by putting special emphasis on the interspecific competition, traffic interactions, and seed dispersal behavior by ants, which are very poorly studied ecology. So we hope, here is today's date, for the first chapter, by the beginning of the next year. Regarding the chapter 2, we have planned another sampling period in this year. We have a prescribed fire in in autumn during this year. And we will do the last sampling period during the spring of 2026. Regarding our third chapter, we have already studied 2 of the 4 wildfires. And during this summer, at the next year's summer, I will be sampling the other 2. And regarding the seed dispersal experiment, I have planned to do them in the same wildfires while I'm doing the sampling period of the chapter 3. I plan to have all my data collected by the end of the summer of 2026, so I can focus in the data analysis and the paper writing during my last year of thesis, and I hope to submit it by the beginning of 2028. So I would like to especially thank all my teammates for their help and the support, and thank you everybody for coming.
So thank you, Juan. It's time for questions.
Thank you. Can you be more specific on the functional diversity of ants? And also, another question, when you say ecosystem function, I mean, besides the seed dispersal, which other one would you consider?
Okay, so regarding the functional diversity, we have selected 13 functional traits which are related with the ant fitnesses, which are, for example, how they start a new colony, if they are a dominant or subordinate species, if there are the number of queens inside the nest, and also which is their feed preference. So there are 13 of them. And the second question was about the functions of ants, right?
No, the ecosystem, when you say that you are also interested to see the effect of the fire on the... When you say that you are interested to understand the effect of the fire, not only on ants, but on their function in the ecosystem?
So as we are evaluating the impact of fires on the some essential services provided by ants, like seed dispersal. We hope this, we hope that results to gather information to try to respond, not to respond, but to have a better understanding about how the ecosystem can behave after that. As for example, in the seed dispersal case, we can know how these Mediterranean shrubs areas compared to the non-burned areas, and whether there is a direction in that type of functioning. Okay. Thank you.
More questions?
Thank you. It's very interesting, and my question comes from the most complete ignorance. So how important, I'm thinking on the prescribed fires mainly, how important is the size and the shape of the fires when you start? I don't know what are the capacity of dispersal of ants, and I was thinking if, compared to a very large fire or a small fire, if that will depend on what it's close by, on maybe what you are looking at when studying the recolonization process. Basically, you're looking at dispersal capacity more than adaptation to a certain landscape features. Does it matter the size of the prescribed fire, or is always a standard? And also, when looking at the natural fires, does it matter if you are looking at the study plots very close to the edge of the fire, or has to be at more than a certain number of kilometers, or how does it work?
Well, first, the size of the fire is important, but it's not as important as the intensity of the fire. So here the main factor will be the intensity, not the size. In fact, the response of ants, it has been proved that varies with this intensity. It is not the same for low intensity fires than high intensity fires. Then, regarding the location of the selected areas, there in wildfires, I guess that it is an important factor. But as we are kind of restricted in this sense, because we are trying to assess how ant responds within a Pinus pinaster forest, which is not very abundant. So we can there is not a lot of plots that match the needs that we want to have in order to try to reduce the variability. But yeah, I think that there is a age effect, because it's not the same whether the forest has been in the center of the fire compared to the edges, because in the edges it can be a colonization process. Meanwhile, in the center of the fire, the response of ant communities is totally different.
If I can relate it to that, are there big differences in the dispersal capacity of these different ant groups?
Yeah. That's one of the functional traits that we are assessing. There are, for example, there are some ants that reproduce inside the nest, and they don't fly. They don't have sexual flights, so they move very close to the origin nest. And there are other species that they present reproductive flights, and they can have larger dispersal distances. Okay.
Okay, so it's time to move to the next speaker. So thank you, Juan.
Thank you to you.
Okay, so Pedro Costales is our next speaker. He is supervised by Xavi Picó here at EBD and José Antonio Mejías at the University of Sevilla. He has been working on his PhD already for 3 years. He has performed some experiments here at EBD in the greenhouses. And he's going to tell us more about the Soncus asper taxonomic complex. So thank you, Pedro, and the floor is yours.
Good afternoon, everyone. So this is the title of my PhD plan, and it's an integrative approach to the diversity of the Mediterranean Soncus asper taxonomic complex. And first, before I begin to tell you about this project, I want to introduce myself and my supervisors. I'm a PhD student working at the Department of Plant Biology and Ecology in the University of Sevilla and in the Faculty of Biology. And I've been doing some research with the genus Soncus for several years now, and in my bachelor degree and master degree with one of my supervisors, which is José Antonio Mejías. And he's a professor at the university with and a taxonomist with many years of experience and relevant contributions to this field, like he's specializing in Asteraceae, and he has contributed to projects like Flora Iberica in genus like Lactuca and Soncus. And my second supervisor is Xavier Picó. As you may know, he's working in this institution, in the biological station of the Llana. And he's an important scientist in the field of evolutive ecology and in the characterization of Arabidopsis thaliana. And here I want to share with you a little bit of background where we are here in this project plan. So we are in the Mediterranean basin, which is one of the multiple biodiversity hotspots of our planet. And it is a really interesting place to study speciation processes and also evolution. And it is an important place for conservation, because here we have around 25,000 species, and at least 50% of them are endemic. And so, however, in terms of taxonomy, and these kind of places present really are hard to characterize, because there are a lot of taxa with taxonomic problems that has to be and we need to give them a solution. That's the case of our study system, and I will talk about it later. And this is the approach of our study. Is a integrative taxonomy approach. It is the thing that has been doing in the last years. Here you can see a graph about the publications of about integrative taxonomy in the databases of Web of Science, and you can see that the popularity of this field has been increasing, and it's about like join several different disciplines like morphology, distribution ecology, genetics, and with all of that made a better and more solid taxonomic studies. So about our study system, I'm studying Soncus asper, which is an annual weed with from the family Asteraceae and with a cosmopolitan distribution. However, its origin has been suggested to be here in the Mediterranean, and it's also a very polymorphic species. That's why in the last revision of this genus, there were described 2 subspecies, and the distinction was made basically by their habit and their life cycle. Here, this one, the subspecies asper, as you can see, it presents leaves all along the stems, and it's also an annual. And the subspecies glaucescens is more subrossulate. They don't have these long leaves in the stem, and it was described as a biennial. However, as I say, it's very polymorphic, and as you can see here in this herbarium sheet, is like an intermediate individual from these two subspecies. These are like the limits and the edges, and this is like more intermediate. It presents leaves in this stem, but they are much shorter than this. And I have to say that most of the references for these subspecies are Mediterranean. However, and revisiting these specimens, there was diversity that has not been recognized when the description of these two subspecies, and that is why in its current circumscription we call the Sonchus asper a taxonomic complex, and we saw that there are two different morphological groups based on the flower sizes. We have here the small flower morphotype, which is composed exclusively by Sonchus asper, and the other one is the large flower morphotypes. And in this group, we include these three species that were described during the 19th century. However, these names are now considered a synonym of the Sonchus asper subspecies glaucescens. So also in the description of the protologs of these names, there are differences in life cycle that are interesting for us for this taxonomic complex. Here you can see them in more detail. We have Sonchus nimani, which is perennial rhizomatous and a Sicilian endemism. Then Sonchus glaucescens, which was described as biennial and is present in the southeast coast of France. It's also a coastal form, as you can see here, the sea, and these are like rock in the cliff. And finally, Sonchus graecus, which is an annual and widely distributed throughout the northeastern Mediterranean. Here you can see more detail their distribution, and this map has been made with occurrence data extracted from localities in the herbarium specimens. And you can see in green Sonchus asper, which is very common in the Iberian Peninsula, but also in the rest of Europe. And Sonchus graecus in yellow is more abundant in the East Europe and Central Europe. And then Sonchus nimani is restricted to Sicily, and Sonchus glaucescens in the southeast coast of France. So this my PhD plan will be divided in 3 chapters. This is the first one, and the main objective of it is explore the ecological, genetic, and reproductive diversity of the Sonchus asper complex. To achieve this objective, we have made, well, four different methodologies, and the first one is the species distribution modeling. We take that map you see before and with the occurrence, and also we take bioclimatic variables and try to predict their potential distribution. Then we also did phylogenetic analysis. We extracted DNA from the natural population, leaves, dry leaves of sample in nature. Then we sequenced that DNA with a rat sequence methodology and ensembled the sequences, and finally inferred the phylogenetic analysis. We also look at the genetic structure of this population using Bayesian clustering, and also a discriminant analysis of principal components. And finally, we tested the reproductive isolation by doing experimental crosses in the greenhouse and obtaining different hybrid lines and study measuring different functional traits, and also then estimating the strength of the reproductive barriers. So here are the results. This is from the species distribution model. Here you can see the distribution for Sonchus asper, Sonchus graecus, and Sonchus laucescens. You can see the distribution for Sonchus nimani because we have low occurrences, so we couldn't model very well that species. And in these maps, the brighter color, like yellows and greens, indicate more presence of that species. And so here, Sonchus asper is more present in the west, yes, in the west Mediterranean, and Sonchus graecus is more present in the East Mediterranean. As you can see here, the differences. And Sonchus laucescens is restricted just to the coastlines. We also predicted the overlapping areas, and here you can see that Sonchus graecus share just 55% of the Sonchus asper distribution. Sonchus laucescens only a 4% of the Sonchus asper distribution, and an 8% of the Sonchus graecus distribution. These are the results of the phylogenetic analysis. Here you can see in this tree that here the outgroups, which are other species of the Sonchus genus, and we can differentiate two clades. This one first clade, which is basically formed by this Sonchus asper, which are all of them small with small flowers, and also that I forgot to say, all of them are autogamous. And here in the clade B, we can see the large flowers group, which are all allogamous. Okay, so this first lineage is composed by only individuals of Sonchus nimani. Then we have here Sonchus graecus, and finally Sonchus laucescens. Here are the results of the Bayesian clustering. In this analysis, we got that the best aggregation of our samples with these two genetic groups. This first group is all autogamous self-compatible individuals, and the other one is the allogamous self-incompatible taxa. Okay, so you can see here also, but however, in the discriminant analysis of the principal components, we can see more or less 4 different groups. Okay, so here is Sonchus asper, here is Sonchus nimani, okay, and then Sonchus graecus and Sonchus laucescens. That's it. And finally, for the reproductive isolation experiment, I have to say that, well, in this graphic we see the contribution to total isolation of pre-pollination barriers in black, then post-pollination barriers in gray, and the potential gene flow in white. So if you follow this color line, you can easily look at whenever we cross Sonchus nimani, Sonchus graecus, or Sonchus laucescens with Sonchus asper, the reproductive isolation of this taxa is greater than if we cross all of them within them. Okay. And here is the second chapter of my PhD plan, and the main objective is to characterize the diversity of the ecological strategies of the Sonchus asper complex. So for that, we will apply this methodology based on the Grimes CSR triangle, which the C indicates competitive, the S stress tolerant, and the R rural. And for that, it's basically you can obtain the percentage of each individual they have of competitive, stress tolerant, or rural by measuring the leaf area, a specific leaf area or leaf dry matter content. And if you measure this, you can put that individual in this triangle. So for that, we have made mainly 2 experiments. This main experiment was made to measure of the ecological strategies. For that, we measure different functional traits, okay? And our number of samples was 438. And then we did an accessory experiment where we measured the competition with another species, which is Bromus. And we did a number of samples of 195. So in this experiment, for doing this experiment, the main experiment, we first take a photo of the seeds that we were going to plant and measure them. Then we follow this germination, and also we follow the growth of the plants by counting the number of leaves every week. Then when enough time has passed and the individual were adults enough, we take an adult leaf, we take a photo of this, and with this photo we can measure the specific area of the leaf. We also weight the leaf in the fresh biomass and the dry biomass. And finally we saw all the specimens, and we have weighed the complete biomass of those individuals. We also have made this impression of the epidermis with a nail polish to count for the stomatal density. And that's it. So the last chapter of my thesis is this one, and the main objective is to evaluate the diversity of Sonchus asper complex using morphometric analysis and then establishing descriptive characters. So for this chapter, we have made a lot of research in herbaria, and we have measured several characters in these herbarium specimens. As you can see here, for example, we have measured the lengths of the basal leaves, also their width, and also, which is an really important character for this kind of group because of their differences in life strategies, we have to take a look at the root part of the plant. We also have measured, for example, glandular hairs, and the seeds. Obviously, the seeds are called achenes, and we have the length of the achene and also their width, and we also take measure of the number of ribs, which are these things, and also the length of the pappus. And finally, which is practically the most important character for differentiate these plants, is these are the flowers of the flower heads, and these are the 2 different morphotypes. Here is the long flower morphotype and the short one, and this is sometimes very difficult because you have to take measure of the ligules and also the anthers, and they are very dry and very stick together, so you have to rehydrate them to try to measure. And that's it. We have revisited a lot of herbarium specimens, and sometimes not all of them you have like the full measure of all characters, because a lot of part can be missing. Like this part, the root part is it's common to be missing in a herbarium sheet, and also sometimes the phenology is not accompanying this, because you can see an individual in flowering but don't have the fruits. And here are some preliminary results. As you can see, but what I'm telling you, there are these two different morphological groups, and based on the ligule length, and you can see this nice bimodal distribution of this character. And that's it. This is finally, to just finish my presentation, past, present, and near future of what we're doing. Here is now, and what we started with the reproductive experiments. Here, meantime, we did DNA extractions. Also, we did a collection trip in Sicily. We did the reproductive and isolation analysis, and also morphometric analysis. We did here in 2024, the last year, these experiments in the greenhouses of the EBD2. And we did herbarium visits to Lyon, Aix-en-Provence, and Toulouse. The other part of the experiment, the competition one. And we plan to, we have been working in the first chapter. We want to submit it by this spring. Then start with the second one and submit it by the end of the year. And finally the last chapter and the defense of the PhD. So I want to thank especially my supervisors, José Antonio and Xavi, also other collaborators like Marcial Escudero and Arnal Marcer, and personal technicians like David Rahel of this institution, and also the staff of the herbarium and the greenhouses of the General Services of Investigation in the university. And thank you all of you for your attention. This is all. Thank you.
Okay, so thank you, Pedro. So is there any question in the audience? Yeah.
Very interesting.
Thank you.
So, especially in the first chapter, you talked about how there was a lot of morphological variation and across the complex, and then you had two species that were widespread and two that were more narrowly distributed. So I was wondering if any component of this morphological variation, or especially the genetic variation, had a geographic component.
Yeah, I think that a lot of there are like a lot of genetic variation has to be because of that ecological geographic component. I don't know. It's
So you had for the two widespread species, they were present in the same place, but some of them were very rare at the edges. But so like the so like the green one that's in Turkey, it's still more genetically similar to.
Here, because I don't. Here we have these samples are from Greece, the Iberian Peninsula, the Italic Peninsula. So all of this is Sonchus asper, and they have obviously they will be genetically different, but not so much. So it's like a thing, you know.
So like you can't. There's not like a group from Spain and a group from Italy and a group from Greece.
Not so much. Maybe there is like a genetic component in these two branches of the phylogenetic tree, but they keep being the same thing, is Sonchus asper, and we could enter in the taxonomic of these different groups, but I don't think they are really different things. I think this is the same one species.
Right. Just like, there's not like isolation with geography.
No, and all of the, like I said, is present these characteristic small flower heads, which are very unattractive to pollinators, and that's why, because this is all autogamous individuals.
Yeah, you can see the branches seem shorter in that clade.
Yeah, the differences between these individuals are very low, I think. Yeah.
Those two SGR1 that sort of hang out there,
Yeah.
are those from a different place or what?
I can't remember right now, but I think these are maybe Italian or from Greece, but I don't know exactly right now.
Oh, I thought you were. Sorry.
Questions?
Okay, thank you very much for the presentation. It's
Thank you.
very interesting. But I have a question regarding the species distribution modeling that you used in the slide. So you mentioned in your talk that because of the few occurrences,
Yeah.
the results didn't, it
No.
was not really good or something like that, you mentioned.
Yeah, but for one of our species,
Yeah. But I wanted just to say that don't mention this in your presentation, in your defense, because then you're basically killing your first chapter. Because the committee would say, okay, if you had this kind of problem, how you're presenting this, But anyway, there are some methods like statistics that you can deal with this problem with the low occurrence data. Have you used any kind of?
no.
Okay.
But
So there are a lot of repetition of the samples, like repeating the simulation and then using cross-validation, bootstrapping ensemble, so you can deal with this. And then another thing, did you use or not?
Sorry?
Did you use any kind of?
yeah, there is some cross-validation between these models, and like there are replicates to do them. So the main issue is that for this, the occurrence data set is that we have like hundreds of Sonchus aspers, hundreds of Sonchus graecus, like more than 50 Sonchus olofesen and only 7 Sonchus nimani.
So you cannot basically model the using 7 occurrence points?
Yeah, we couldn't model that.
Yeah.
That's what I was saying. So, but I don't think it's really a problem, because we know certainly that is only here in Sicily, and maybe, but it's like because we are only modeling like the north slope of the Mediterranean basin. Maybe Sonchus nimani is in the north of Africa, but we don't certainly know. But we have been here and we couldn't find any Sonchus nimani in the Italian peninsula.
Okay, and then for the you did some overlap.
Yes.
No? And then how did you do that? I mean, you did some kind of measurement, some kind of similarity between a species, or any kind of metrics for the similarity?
I don't know exactly, but I think it's just like modeling this, unifying the models.
Well, okay, but this is not that much correct. So you have two options. First of all, you can use threshold-based models, like based on one of the threshold metrics, to have 0-1 presence absence of a species, and then combine this. This is also possible.
Yeah, we have to do this 0-1.
Or doing like one of the similarity metrics that they use for the species, finding a habitat similarity of both species, and give you the percentage of the overlap for the habitat, or for geographic or environmental, both of them. We can also calculate this, which is very important to mention, I think, because, okay. Thank you.
Thank you.
Okay, maybe one short question. Okay.
Thank you very much for the presentation. I think I've missed it, but what markers have you used for your genetic analysis?
Markers? Well, we have sequenced the dry silica dry leaves with rat sequence, like restriction site associated DNA sequences, and then we have used that the reads the sequence for for inferring the phylogenetic analysis, and for the genetic clustering we have used SNPs, the dataset of SNPs, one randomly selected variable locus.
And the approximate number of SNPs that you found, more or less?
More than, I think it's 13,300, something like that.
Okay, so it's 5 minutes after 2. So thank you very much all for coming, and thank you for your questions and comments. I think that they're going to be very useful for them. Thank you.